Today's module is all about the cell. The cell is the basic unit of life, and so we might as well get right into it. As you probably know, cells are really small, and one of the reasons that they're small is that they need to maintain a certain ratio of surface area to volume. So let's go ahead and imagine that the cell is a cube, and if we do that then, and we want to determine surface area of this cube, we would multiply height times width, so x times x would be x squared. And that would give us the surface area for one of the surfaces. However, we would have 6 surfaces, so we would need to multiply that times 6 to get the actual surface area. So if we imagine the surface area to be 1 millimeter, again 1 times 1 is 1, that would be x squared. 6 times that, because we have 6 sides, would give us 6 millimeters squared for the surface area of this cube. Now let's compare that then with volume. In volume we would multiply the height times the width times the depth. And in this case, if they're all equal to 1, so let's assume a cube of 1 millimeter again, if we did that, we would get a volume of 1 times 1 times 1, which would be 1 millimeter cubed. So now if we look at the ratio of surface area to volume, again for a 1 millimeter cube, then that ratio would be 6 to 1, or a total of 6. Now, let's compare that to a larger cube. And the problem here, and the reason that surface area is so important, is let's say we have a large cell. What needs to happen is that particles have to diffuse from the surface into the interior of this cube, or into the interior of the cell. So if you have a large cell, molecules have to diffuse a very long distance. And if you have a small cell, it's a much shorter distance that they actually have to diffuse. And again, as we're going to see in a second, you'll have more surface area for unit of volume here than we will here. So let's go ahead and compare then a large cube to a smaller cube. So we just computed surface area and volume and the surface area to volume ratio for a 1 millimeter cube. Now let's say that we had a larger cube that was 5 millimeters or 5 units, it doesn't really matter what they are, on these axes. In that case then, what we would have then is a surface area of 150. We would have a total volume of 125. And so now our ratio of surface area to volume is much less. So in other words, there's less surface area for molecules to diffuse, and they would have a longer distance to diffuse to get into the interior of the cell. So again, a way to get around this then is what if we took that 5 millimeter cube or 5 micrometer cube, whatever it is, and divided it up into 1 millimeter cubes. If we did that, then if we looked at the total surface area, it would be 750. The volume is still 125, but that gets us back to this 6 to 1 ratio. So again, one of the ways that cells get around being too large is just to divide and make sure that they stay small to maintain this optimum surface area to volume ratio. Okay, now there are other adaptations. So adaptations to maintain a high surface area to volume ratio, certainly one is to be small. Also having highly folded membranes, cellular membranes on the surface especially, will increase surface area relative to volume. Also being broad and flat would be another adaptation that could do this as well. So again, the idea is to minimize the distance between the surface and the interior of the cell. In that way, chemical reactions can occur at a rapid speed. Now, when we look at cells, one of the tools that we use is the microscope. And there are a number of different microscopes. We're going to look mainly at light microscopes, which rely on light to either pass through a specimen or bounce off of a specimen, and also electron microscopes, which use electron beams. There are many other types of microscopes, but again, we're going to keep it kind of simple here. Now, when we look at microscopes and microscopic power, one of the ways that we define them is by means of what's known as resolving power. Now, resolving power is a measure of the minimum distance at which you can distinguish between 2 points. So, for example, if you have 2 structures that are separated from one another, clearly you can distinguish my 2 fingers are a certain distance apart. But as I move them closer and closer together, there's a point at which it's going to be difficult for you to tell whether my fingers are touching or not. Okay, that minimum distance at which you can still determine that my fingers are not together, that's the resolving power. And there is a relationship between resolving power and magnification. So in other words, if it was a distant shot and I had my fingers close together, you might not be able to tell whether they were together or not. But if the camera zoomed in, you might still see that there was a space between them. So generally speaking, as magnification increases, the resolving power distance decreases. However, it is dependent upon the quality of the lens. In other words, if you magnified something greatly, but it was a blurry lens, you still might not have a very good resolving power. So again, generally there's a relationship. As magnification increases, resolving power gets smaller, the distance gets smaller, but it depends on the quality of the lens. Okay, so with the naked eye then, our resolving power is about 0.1 millimeters. And a millimeter is 1/1000th of a meter. And so we refer to that magnification as being 1x. So just to give you that as a reference point then, as we look at the quality of light microscopes, the very, very best light microscopes have a resolving power of 0.2 micrometers. And a micrometer is a millionth of a meter. And they can have magnifications up to 2000x. And then comparatively, electron microscopes have a resolving power of 2/10,000ths of a micrometer, 2/10,000ths of a millionth of a meter. And they can have magnifications up to 1,000,000 times. So they are much more powerful, but much more difficult to use, obviously. Now, in looking at cell types, there are two major types of cells in nature, prokaryotic cells and eukaryotic cells. And so we're going to begin with prokaryotic cells. The term prokaryotic is trying to tell us that karyo is a reference to a nucleus or core, and pro means before. So these are organisms that evolved before a nucleus. So they lack a true nucleus. And what defines a true nucleus is being a membrane-bound organelle that contains the genetic material. So they lack this true nucleus, and we'll get to that example later. They also lack membrane-bound organelles. That won't mean much right now, but we'll see lots of those when we talk about eukaryotic cells. They also tend to have prokaryotic ribosomes and flagellae. So they have ribosomes and flagellae that are similar to those in eukaryotes, but unique to the prokaryotes. And generally, prokaryotes are just very small. So typically less than 10 micrometers. A typical size, however, is around 2 micrometers, 2 millionths of a meter. So let's go ahead now and look at prokaryotic structures, basically working from the outside in. So first, a capsule. The capsule is the outermost structure, and it's not found on all bacteria, but many bacteria. And when present, the capsule is composed primarily of lipids. It can have other compounds associated with it. And in some species of prokaryotes, this is associated with disease. And I should mention, too, that prokaryotic organisms include things like bacteria and a group known as the archaeans. So, and they're essentially ubiquitous in all ecosystems. Some of them cause disease. So many disease-causing bacteria, for example, then will have a capsule associated with them. And so sometimes the capsule itself may have toxic elements, but the capsule also is kind of slimy, and it is protective in some situations against our immune cells. So whereas we have white blood cells that would try to be eating the bacteria, the capsule makes them kind of slippery, and it's like trying to pick up a bar of soap. It'll kind of squirt out, and they think that's one of the ways that the capsule works. Now, when the capsule is present, again, it would be the most outermost structure. Now, another structure that we see, or structures that we see associated with the cell wall, sometimes with the capsule, are what are known as fimbriae or pili. And these are just extensions from the cell wall. And these can have a couple of functions. Fimbriae tend to attach prokaryotic cells to other prokaryotic cells. It can attach them to surfaces. And then also some of the pili would be plural, pilus is singular. Some of these are involved in sexual processes. So a pilus will grow between one bacterium and another, for example, and then that can allow for the transfer or exchange of genetic material. And this is one of the problems with bacteria, is that they are capable of exchanging genetic material with very unrelated bacteria, sometimes even with a group known as the archaeans, which we'll talk about later. And so anyway, they can swap genes back and forth. This is one of the problems that we're having with bacteria becoming antibiotic resistant, because an antibiotic resistant gene can easily be swapped to another bacterium. All right, now the cell wall then would be internal to the capsule, if a capsule is present, and the cell wall is highly permeable. And in many prokaryotes, the cell wall is composed of a material known as peptidoglycans. And so the peptido is telling us that there's a protein element here, or amino acid element. Glycans is a reference to sugars. And so we sometimes refer to these as mucopolysaccharides, but peptidoglycans are kind of slimy, a combination of protein or amino acids and sugars. And again, we consider the cell wall to be highly permeable. It is not selectively permeable. Particles move through it quite readily. And it helps maintain the shape of the prokaryote. In some cases, actually, the cell wall may have toxic elements if it's a pathogenic or disease-causing type of prokaryote. Internal to the cell wall, then, is really the beginning of the cell, in a sense, the living organism, and this would be the plasma membrane. We've already talked about plasma membrane, so again, in a prokaryote, we're looking at the same basic structure of the cell membrane, a bilayer or double layer of phospholipids with different types of proteins embedded in that membrane. And the cell membrane is the semi-permeable barrier from the outside world. Now, associated with the cell wall and the cell membrane in many flagella, in many types of prokaryotes are flagellae. And so the prokaryotic flagellae is different than the eukaryotic flagellum that we'll talk about later, but the flagellae help propel the organism through the water, and generally they pull it through the water. And so where the flagellum attaches to the cell wall and the cell membrane, there's actually kind of a bushing there that allows it to spin around. So the flagellae here spin, and that's what pulls the bacteria through the water. So if they lack that, they would have to have either they're not motile or they'll have some other types of mechanisms for moving. But flagellae are all about movement. And the word flagellum really comes from whip. So flagellae are whip-like structures associated with the cell. Now, internal to the cell membrane, then, we would consider that the cytoplasm of the cell. And that's not highlighted here, but just that's a general term. Now, within the cytoplasm, the most dominant structure, if you want to call it that, is what we call the nucleoid. And the nucleoid isn't a nucleus. It just means it's nucleus-like, and really what it's composed of is the genetic material of this prokaryotic cell. So all of the genetic material then is compacted into this central core known as the nucleoid, and it's concentrated, as we can see here on the diagram. Now, prokaryotes have a single large circular chromosome, and we also refer to it as being a naked chromosome, and by that I mean there are no protective proteins associated with it. It's just a DNA double helix. So that's the largest genetic structure within bacteria. Additionally, there are smaller circular rings known as plasmids. So that constitutes the genetic material in a prokaryotic cell. Additionally, in the cytoplasm, there are small structures known as ribosomes. Ribosomes make proteins, and we'll learn a lot more about those later. Now, one last kind of structure we may see in prokaryotes is that some prokaryotes will have a number of internal membranes. And these are referred to as photosynthetic membranes if they're associated with some type of photosynthesis, respiratory membranes if they're involved in producing ATP. So anyway, we'll talk more about those processes later in the course. Now, eukaryotes. You and I, for example, are eukaryotes, are distinguished by the fact that they possess a true nucleus. And a true nucleus is encapsulated in a nuclear envelope. The nuclear envelope is composed of two plasma membranes. Also, the chromosomes within the nucleus are linear rather than circular. Eukaryotes possess membrane-bound organelles, as we'll see in a second, and they have distinct eukaryotic ribosomes and flagellae, and they tend to be big, almost always larger than 10 micrometers. Typically, a typical size would be 100 micrometers compared to 2 micrometers in prokaryotes. All right, so first let's look at an animal cell and look at the structures associated with an animal cell. And so one thing to notice is that animal cells do not have any kind of a cell wall, so the outer boundary of the cell will be the plasma membrane. And again, the plasma membrane is going to be our typical membrane structure, a bilipid, biphospholipid layer with proteins embedded. This is the semi-permeable barrier between the outside world and the inside world. And again, typically there will be a lot of proteins associated with the cell membrane, extracellular proteins and intracellular proteins, many of which form a cytoskeleton, a cell skeleton. And the cytoskeleton lends substance to the cytoplasm, the material inside the cell membrane. Also, they can be used as pathways to move materials from one place to another, kind of like railroad tracks. Now, again, the dominant feature in a eukaryotic cell is the nucleus. And so the nucleus contains material called chromatin. It contains a structure known as the nucleolus, and the nucleolus is a concentration of RNA. And also the nuclear envelope then is the outer boundary of the nucleus. And again, the nuclear envelope isn't a single plasma membrane, but two plasma membranes. So if we look at the structure of the nucleus in a little more detail, again, it would have two plasma membranes rather than a single plasma membrane. Again, chromatin and a nucleolus composed of RNA. Now, this chromatin then is really what the chromosomes are, and they're all compacted together. So when we look at the structure of a chromosome then, the hereditary portion of a chromosome is the DNA double helix, and it will wrap around and associate with proteins, and then those will compact into a chromosome. So this combination of DNA and protein, we refer to as chromatin. But what I do want you to know at this point is that the genetic component of chromatin is the DNA. So chromatin is the material of which chromosomes are made, and chromatin is composed of the DNA double helix and proteins that it wraps around, essentially. And again, this chromatin then coils up and forms the chromosomes. And again, we have linear chromosomes. And in our normal cells, human cells, we have 23 pairs of chromosomes, so a total of 46 chromosomes. In our normal body cells, our sex cells will only have 23 chromosomes. Now, associated with the nuclear envelope is a number of highly folded membranes, which create a series of channels that go through the cytoplasm. And these are known as the endoplasmic reticulum. And so there are essentially two types of endoplasmic reticulum. There's a rough endoplasmic reticulum and a smooth endoplasmic reticulum. And the endoplasmic reticulum is connected to the nuclear envelope. So the outer membrane of the nuclear envelope folds out and forms this incredible network of membranes, which forms the endoplasmic reticulum. If proteins are associated with the if ribosomes are associated with the endoplasmic reticulum, we call it a rough ER because it looks grainy in electron microscopy. If there are no ribosomes associated with it, it's called the smooth endoplasmic reticulum. Now, endoplasmic reticulum then, if there are ribosomes associated with it, you may recall ribosomes make proteins. So this is where a lot of protein production occurs within the rough ER, and then the rough ER is continuous with the smooth ER, so some of these proteins then move into the smooth endoplasmic reticulum. Now, as I just mentioned, ribosomes make proteins and will typically be associated with the endoplasmic reticulum, but they can also be free in the cytoplasm. Ribosomes have two subunits to them, but we'll learn a lot more about those later in the course. Golgi apparatus is a series of coin-like membranous sacs, and they also associate with endoplasmic reticulum. And so what happens is that the products from the endoplasmic reticulum will pinch off, form vesicles, and fuse with the Golgi apparatus. And then this material is, these proteins then are modified in the Golgi apparatus and eventually form other vesicles which either become organelles within the cytoplasm, or in some cases these materials actually are moved to the cell membrane and secreted outside the cell. So cells that produce a lot of secretory products like hormones and so on will have lots of Golgi apparatus, and they will move that material out of the cell. Now, lysosomes are structures that are actually produced by the Golgi, and lysosomes contain digestive enzymes. So lysosomes will associate with typically food vacuoles. If a cell brings in some large particle in a vacuole, then lysosomes can associate with those and digest that material. Lysosomes can have some other functions as well. For example, when you die or when a cell dies, or if it's programmed for cell death, the lysosomes can rupture, and that begins the digestive process and decay process of a body or of a cell. Now, mitochondria are still other organelles, and these are important in producing cellular energy, a type of energy known as ATP, and we're going to learn a lot more about that. What I would like you to know about them now is that they do have two membranes, an outer membrane and an inner membrane. The inner membrane is highly folded. And mitochondria evolved from symbiotic bacteria living within eukaryotic cells. So they have their own complement of DNA, they have prokaryotic type of ribosomes, and they're capable of dividing independently of the cell. So ribosomes, we're pretty confident, evolved from prokaryotes that were living inside of eukaryotic cells. Now, other organelles, a general term is microsomes. Microsomes are tiny vacuoles that contain specific enzymes. A peroxisome is one that contains an enzyme called catalase, which counteracts the effects of hydrogen peroxide, which is potentially toxic to cells. And microsomes are also generated by the Golgi apparatus. And then centrosomes are composed of two centrioles at right angles to one another. And the centrosome is responsible in animal cells for cellular reproduction, so they play a role in that. And plants lack centrosome or centrioles. Additionally, vacuoles are just large sac-like structures, membrane-bound structures. They can be used for storage of various materials. The term vacuole means if it's a large membranous sac. Vesicle means a small membranous sac. And there are some vacuoles called contractile vacuoles that are specialized for pumping water out of a cell. So for example, a unicellular organism living in fresh water, it's hypertonic to the surrounding water. Water moves in, it fills up this contractile vacuole, and that vacuole will contract and basically squirt that water out of the cell. And then the flagellum or cilium is a mode of movement. And so flagella and cilia have the same internal structure. It's just that if it's a long structure, if it's whip-like, it's a flagellum. If it's more hair-like, it's called a cilium. And so again, these would be examples of flagellae. Here would be the cilia. And in cross-section, they're identical. They're composed of 9 doublets of microtubules with a central core of a doublet of microtubules. And then at the base of those, there's a structure that's actually identical to the structure of a centriole, and it's called a basal body. And the basal bodies control the function of the cilia or flagella, although there's a lot of question as to how they do that. And one of the differences in mechanisms is that instead of a spinning type of flagellum, so in the prokaryotic cell where it spins around, the microtubules in eukaryotic cells slide over one another. They're little motor molecules, and as these tubules slide past one another, then that's what causes bending and movement of fluids over or movement of the cell itself. Okay, now plants basically have all of the structures we've talked about with a few exceptions, and I'll get to those exceptions in a second, but plants have a cell wall external to the cell membrane. And again, the cell wall would be highly permeable. The plant cell wall is composed of cellulose, and we've talked about cellulose before, a polysaccharide. So, however, and the cellulose, remember, is composed of glucose. So the cell wall provides some structure to the cell. It's not extremely rigid, but it provides some structure to the cell, and again, highly permeable. And that would be external to the cell membrane. Plants also have chloroplasts. We kind of generically refer to them as plastids, and so plastids will have 2 membranes, an outer membrane and an inner membrane, much like mitochondria. And we again think they evolved from prokaryotic organisms that were living inside of eukaryotic cells. So they also have their own complement of DNA and can divide independently of the cell. So when you look at plastids, there are 3 types. Chloroplasts contain chlorophyll, and they're involved in photosynthesis. Chromoplasts you would find in flower petals. They contain pigments. And leucoplasts we essentially are modified chloroplasts that you find in roots. Plant can't photosynthesize when it's underground, or the roots can't photosynthesize underground. So the leucoplasts store starch. And leuco means white, chromo means colored, chloro is a reference to chlorophyll, green. Okay, plants also typically have a large central vacuole, fluid or water-filled vacuole called a tonoplast. And then also plants will have connections between cells called plasmodesmata. And so the plasmodesmata, again, are actually openings from one cell to the other through the cell wall, so that the cytoplasm of one cell is continuous with the cytoplasm of another cell. So they actually share cytoplasm. So when we look at animal cells then, animal cells lack a cell wall, they lack plastids, they lack plasmodesmata, and they possess centrosomes and centrioles. Plant cells, again the other major category of eukaryotic cells, possess a cell wall, possess plastids, possess plasmodesmata, but lack centrosome and centrioles. Now, the last thing I want to mention briefly, and I know I've gone very fast today, I'm sure you'll have to stop and replay some of what I've talked about. But when we look at cells and try to figure out what the cellular organelles do, the membrane-bound organelles do, especially in eukaryotic cells, What we do basically is take a sample of the tissue, run it through a blender, and basically break up the cells, and then that produces what we call homogenate. And then we will take this homogenate and put it into something called a centrifuge. And a centrifuge is something that spins very, very rapidly. And what will happen is that materials that are suspended in the homogenate will be driven to the ends of the test tube. And so what will happen then is we do this at different speeds. So we'll take this homogenate, run it through a centrifuge, it'll create a pellet, and then we'll pull that pellet out and try to figure out what the structures are and what those structures do. Then we'll take that supernatant, put it in another tube, and run it through a centrifuge at a higher speed and for a longer period of time. That will pull out still smaller structures into a pellet. We'll take those pellets out and examine them and try to figure out what they are and what they do. That supernatant again can be centrifuged again. And so this is known as cell fractionation. So what we do then is break up the cells and then separate out different parts of the cells and then try to characterize them in terms of their structure and in terms of their function. Now, very quickly then, when we look at organisms, some cells are composed, of one cell, they're unicellular. Some cells are loose aggregations of cells, we refer to them as colonial. And still other organisms like you and I are multicellular. So our individual cells can't go off and live by themselves, whereas if you're colonial, you can do that. And then also, multicellular organisms, the cells combine in many cases to form tissues with specialized functions, and then tissues of specialized function in turn form organs. So bottom line then is that all organisms are composed of cells and their cell products, and cells are derived from previously existing cells. So that's what's known as the cell theory. Now, many of our tissues are composed of extracellular material. So for example, if you look at our tendons in our cartilage, there are actually relatively few cells there. Many times there are actually cell products that are secreted outside the cell, and that will make up the bulk of a tissue. So again, as you look at our cartilage, look at our tendons, our ligaments, things like that, most of that is what we would refer to as extracellular material. So I know I've gone really fast today. So again, it's going to be important to go ahead and stop this lesson when you can, stop this module when you can, go back and listen again and use your other materials. So we've taken a really rapid tour of prokaryotic and eukaryotic cells. So until the next time.